We've delved into the details of the catabolic reactions of cellular respiration in which aerobic organisms oxidize organic compounds like glucose, obtaining energy to synthesize ATP. Now we'll turn our attention to photosynthesis, which of course is the source of those organic compounds. Photosynthetic organisms are able to transform light energy from sunlight into chemical energy in the form of simple organic compounds built from CO2, carbon dioxide, and electrons from water. Since they're making their own organic compounds, photosynthetic organisms are one type of autotroph, or self-feeder. Organisms like us that can't do that and have to steal food from others are referred to as heterotrophs. When thinking about photosynthetic organisms, usually what first leaps to mind are the land plants that we see all around us, but we should recognize at the outset that they are not the only photosynthetic organisms. Marine algae, protists, and cyanobacteria all carry out similar oxygenic photosynthesis reactions, in which sunlight energy is captured, water is oxidized, yielding oxygen, and carbon dioxide is captured from the atmosphere and reduced to form simple sugars. As we've discussed before, other prokaryotic organisms, like the purple sulfur bacteria we see here, carry out anoxygenic photosynthesis using other electron donors besides water. We should also recognize that in terms of global carbon capture, the marine algae, cyanobacteria, and protists are just as important as land plants on a global scale. As we've discussed already in the course, chloroplasts are the organelles responsible for carrying out photosynthesis in eukaryotic organisms. If we zoom in on these little compartments and examine their structure in more detail, we see that, like the mitochondria we've already talked about, chloroplasts also have an inner and outer membrane. But in addition, chloroplasts have another set of flattened, membrane-enclosed compartments inside the inner membrane, and these are called the thylakoids. Usually these are stacked one on top of the other, forming what's called a granum. The interesting light energy capturing reactions of photosynthesis are going to involve pigments and other molecules embedded within these thylakoid membranes, so we can see the advantage of having this extensively folded membrane structure. It increases the surface area for these pigments and proteins, and therefore increases the amount of photosynthesis that each chloroplast can carry out. Here's an electron micrograph showing what a chloroplast really looks like, and you can see the grana, the stacks of these disc-like thylakoids. The two compartments that are going to be particularly important for the processes and reactions of photosynthesis are the interior of the thylakoids, called the thylakoid lumen, and the space outside the thylakoids but inside the inner chloroplast membrane, called the stroma of the chloroplast. You might notice that there are connecting tubules between the grana, so this thylakoid space or lumen is really one continuous compartment inside the thylakoids. Just as we saw with cellular respiration, the reactions of photosynthesis are redox reactions, and the summary equation for the process is shown here at the top. Using the energy from sunlight, 6 carbon dioxide molecules are reduced to form sugar, and this is linked to the oxidation of water yielding oxygen as a waste product. Now this is a summary of what happens overall, but as we'll see, the process involves a number of different steps. But overall, we can divide the process of photosynthesis into 2 general stages. In the light reactions, what we might consider to be the photo part of photosynthesis, light energy from sunlight is captured and converted to chemical energy in the form of ATP and a reduced electron carrier coenzyme, in this case, nicotinamide adenine dinucleotide phosphate, or NADP, which is nearly identical to the NAD coenzyme you were introduced to in the cellular respiration discussion. It's just phosphorylated. Just as we saw with NAD and FAD, NADP can be reversibly reduced and oxidized, and so effectively it can act to shuttle electrons from one molecule to another. And as we've talked about a lot, ATP is used as energy currency. And in this context, the synthesis of ATP using energy from sunlight is going to provide a cellular energy store, which can then be coupled to synthesis of sugar in the next stage of photosynthesis, the Calvin cycle, which is where carbon dioxide is reduced using the energy from ATP and NADPH to form sugar. So this would be the synthesis part. The light reactions involve light-absorbing pigments and proteins embedded in the thylakoid membrane. whereas the enzymes that catalyze the reactions of the Calvin cycle are in the stroma of the chloroplast. So again, what we see happening is that light energy from sunlight is absorbed and converted by the light reactions to chemical energy in the form of reduced NADPH and ATP. The reduction of NADPH is linked to the oxidation of water, which yields molecular oxygen as a waste product. The ATP and NADPH energy is used in the Calvin cycle to convert carbon dioxide to sugar. More specifically, the 3-carbon sugar glyceraldehyde 3-phosphate, or GAP, which, as you'll recall, was one of our intermediates in the breakdown of glucose during glycolysis. Once ATP is hydrolyzed and NADPH is oxidized in the Calvin cycle reactions, the resulting ADP and phosphate and NADP+ are made available to the light reactions once again to regenerate ATP and NADPH by the light reactions as long as light is available. This provides the energy input the enzymes of the Calvin cycle need to reduce carbon dioxide. We're going to look at both of these stages of photosynthesis, beginning with the light reactions. Before we talk about the light reactions, we need to talk a bit about light itself. When we say light in this context, we're really talking about visible light, that part of the electromagnetic spectrum between, say, around 400 and 700 nanometers in wavelength, although we should recognize that there are organisms that are able to use infrared and ultraviolet wavelengths as well. Remember that wavelength corresponds to energy level. The shorter the wavelength, the higher the energy level, and the longer the wavelength, the lower the energy level. So the wavelengths in the blue and purple end of the spectrum have more energy than the wavelengths towards the red end of the spectrum. Because they are at different energy levels, these wavelengths of light are going to interact differently with the pigment molecules that are absorbing this light energy, with specific pigments absorbing some wavelengths of light, but not others because of their molecular structures. Here we see the molecular structure of the main photosynthetic pigment found in all organisms that carry out oxygenic photosynthesis, chlorophyll A, here on the left. Chlorophyll A has this characteristic large ring structure with a coordinated magnesium ion in the center. This is referred to as a porphyrin ring. Because of its specific molecular structure, this chlorophyll will interact with specific energy levels, specific wavelengths of light, and electrons will be excited from ground state to excited state. Remember from chemistry that this excitation energy has to match the quantum energy level required to bump the electron up to the excited state. Too much or too little energy won't work. For chlorophyll A, the energy levels that work best are those in the red and blue ends of the spectrum, but not the green wavelengths. Different pigment molecules with different molecular structures will interact with different energy levels of light, and so will absorb or capture different wavelengths. These other pigments are referred to as accessory pigments that may or may not be found depending on what organism you're looking at. There are other chlorophyll molecules with very similar but distinct porphyrin ring structures, including chlorophylls B and C. Phycobilins like this one, phycoerythrobilin, are common in cyanobacteria. Fucoxanthin is found in the brown algae like the kelp we see off of our coast. And beta-carotene is commonly used as an accessory pigment in lots and lots of different organisms. The advantage of having more than one type of pigment is that it broadens the wavelengths of light that can be used for photosynthesis. If we compare the absorption spectra of different photosynthetic pigments, in other words, the wavelengths of light that they can interact with and absorb, we see that they are very specific. Here's the absorption spectrum for chlorophyll A in light blue, and you can see that this pigment absorbs maximally at around 420 nanometers and 670 nanometers, whereas the chlorophyll B molecule, which is almost identical structurally, absorbs maximally at around 450 and 650 nanometers. So if an organism has both chlorophyll A and chlorophyll B, it's capable of using more of the visible spectrum for photosynthesis. In other words, it's more efficient at capturing light energy for photosynthesis. Plants primarily use chlorophyll A and B and carotenoids, So the wavelengths absorbed are between around 410 and 470 nanometers in the blue end of the spectrum, and between about 640 and 680 nanometers in the red end of the spectrum. But notice that those pigments really don't have any absorbance of the wavelengths corresponding to green here in the middle. So because those wavelengths pass right through without being absorbed, or are reflected off, what we see when we look at a plant is green. The other wavelengths of light are being absorbed by the plant pigments. Cyanobacteria, on the other hand, might have these phycobilin pigments in addition to chlorophyll, and so they would capture more of these wavelengths in the green and yellow regions of the spectrum. Okay, so pigments have specific molecular structures that allow them to absorb specific energy levels or wavelengths of light. How does that captured energy then get transformed to chemical energy? To answer that question, we have to look in more detail at the structures in which the pigment molecules are embedded, called photosystems. Photosystems are large complexes made up of many proteins, represented in purple, and pigment molecules, represented in green, that are located within the thylakoid membranes of the chloroplast. Most of the pigments are located in a region of the photosystem complex called the light harvesting complex, which is illustrated in darker purple in the diagram. But there's also a special pair of chlorophyll molecules located in an inner region of the photosystem that's called the reaction center, highlighted in a lighter purple. This special pair of chlorophyll molecules is special not because of any structural difference in the pigment molecule itself, but because of its molecular neighborhood. More specifically, it's located right next to a molecule called pheophytin that can strip away excited electrons from that pair of chlorophyll molecules, and this is called the primary electron acceptor. So what happens in the photosystem is that light energy is being absorbed by all these pigments in the light harvesting complex. Chlorophyll A and all those accessory pigments are absorbing their particular wavelengths of light that they're sensitive to, and when hit by those wavelengths, electrons in those pigment molecules are being raised from the ground state to the excited state. These pigment molecules then pass around that excitation energy between themselves in the form of resonance energy transfer. So let's say we have an electron in one pigment molecule raised to the excited state by energy absorption. When it falls back down to the ground state, it passes off that excitation energy to a neighboring pigment molecule, which raises its electron to an excited state. Note that in this case, no electrons are being transferred yet. Nothing has been oxidized yet. It's only excitation energy being transferred, not electrons. But when that excitation energy is finally transferred to the special pair of chlorophyll molecules in the reaction center, raising its electrons to the excited state, those excited electrons do get stripped away from the chlorophyll by the primary electron acceptor nearby. So the special pair of chlorophyll become oxidized, and the primary electron acceptor becomes reduced. It's at this point that light energy has been transformed into chemical energy by driving the transfer of electrons from the reaction center chlorophyll to the primary electron acceptor. Now it's fine to represent a photosystem as this purple blobby thing with green pigment spheres, but I want to show you what a real photosystem looks like in all its magnificence. Here's the structure of a real photosystem, which would be embedded in the thylakoid membrane. We're looking at it from the side. So in here would be the interior of the thylakoid, or thylakoid space, or lumen. This is where the thylakoid membrane is, and out here would be the stroma of the chloroplast. You can pick out the transmembrane alpha helices, some more alpha helices here, and beta sheets of the protein component of the photosystem. And if you look closer, you can also see the structures of the pigment molecules embedded within the protein structure. Here's another view from the top down, omitting all of the protein and just showing the arrangement of the pigment molecules themselves. In this particular photosystem from cyanobacteria, we have 3 light harvesting complexes surrounding 3 reaction center chlorophyll special pairs. And you can see how closely juxtaposed these pigments are relative to one another, allowing them to transfer that excitation energy among themselves and ultimately to the reaction center chlorophyll. Much more impressive than that purple blob, but the blob is useful when we're simplifying to get an overview of what's happening. Okay, so far, light energy has been absorbed, passed around between pigments, and ultimately the reaction center chlorophyll were oxidized, and the primary electron acceptor was reduced. What happens next? To understand what happens next, you need a bit more detail on the photosystems and the other molecular components in or near the thylakoid membrane. We're looking at a representation of a thylakoid here. The thylakoid membrane is in gray, and here is the thylakoid space, and outside is the stroma of the chloroplast. In the membrane, we would find two different types of photosystems, called Photosystem 1 and Photosystem 2. They have essentially the same structure, protein and pigment, light harvesting complex surrounding a reaction center core. What's different about them is how they replace lost electrons. In other words, when the reaction center chlorophyll gets oxidized, where do the electrons come from to replace those lost electrons? In addition to the photosystems, there's an electron transport chain consisting of a cytochrome complex, called cytochrome B6F, and membrane-associated mobile electron carriers, which can move within or along the membrane and facilitate the transfer of electrons between the photosystems and the cytochrome complex. An enzyme called ferredoxin NADP reductase is found in association with the stroma side of the thylakoid membrane, and this enzyme, as its name suggests, catalyzes the reduction of NADP to NADPH. Also found in the thylakoid membrane is ATP synthase, the very same ATP synthase protein complex that we discussed as the main source of ATP in mitochondrial oxidative phosphorylation. Its presence and that of an electron transport chain complex might give you a hint as to what we're going to see happening here. Transfer of electrons, pumping of protons, chemiosmosis driving ATP synthesis, and you'd be exactly right. The mechanism is identical to what we saw happening in the mitochondrion with oxidative phosphorylation. In this case, since energy to move electrons is being derived from sunlight, here the mechanism is referred to as photophosphorylation rather than oxidative phosphorylation. Okay, so let's put all these pieces of the puzzle together and see how they might act together to transform sunlight energy to chemical energy in the form of ATP and reduced coenzyme NADPH in a process called linear or non-cyclic electron flow. Both photosystems are bombarded with light energy, and pigment molecules in the light harvesting complexes will absorb that light energy, exciting the pigment molecule electrons. Resonance energy transfer is going to occur, with the energy ultimately being transferred to the reaction center chlorophyll molecules, which become oxidized. Now, the same thing is happening in both photosystems, but let's zoom in over here at photosystem 2 and see what happens next. Chlorophyll is oxidized when its excited electrons move to the primary electron acceptor. From there, the electrons move to a mobile electron carrier in the thylakoid membrane. Now, once that happens, the chlorophyll in the reaction center becomes an incredibly powerful oxidizing agent. It's looking to replace those lost electrons, and it's such a powerful oxidizing agent that it's capable of oxidizing water, and that's exactly what happens. To replace two lost electrons, two molecules of water are oxidized, yielding molecular oxygen and 4 protons. The oxygen diffuses out, and the protons contribute to a proton gradient across the thylakoid membrane. Incidentally, this is the only example of enzymatic oxidation of water that we know of. Okay, so the chlorophyll has replaced its lost electrons, but what happens to the electrons that got passed from chlorophyll to the primary electron acceptor and from there to the mobile electron carrier? Those electrons are going to be passed along the electron transport chain and ultimately are going to be passed to the reaction center chlorophyll in photosystem 1, which has also been oxidized. Remember that while photosystem 2 was being stimulated by light energy, the same thing was happening over here at photosystem 1. In linear electron flow, the electrons lost by the reaction center chlorophyll of photosystem 1 are passed to the primary electron acceptor, then to a mobile electron carrier, a protein called ferredoxin, and from there, the enzyme ferredoxin NADP reductase oxidizes the ferredoxin and transfers the electrons to NADP, forming NADPH. So once again, in linear electron flow, electrons flow from oxidized water through photosystem 2, down the electron transport chain, through photosystem 1, and ultimately end up in NADPH. As electrons are moved down the electron transport chain, this cytochrome B6 complex is going to transfer protons from the stroma into the thylakoid space, building a proton gradient, a proton motive force, just like what happens in the mitochondrion. Just as we saw before, that proton gradient is the direct energy source for ATP synthesis by the ATP synthase enzyme complex. As chemiosmosis occurs and protons flow through ATP synthase down their concentration gradient, the rotor turns. This internal rod turns within the catalytic subunits, changing their shape in such a way as to force ADP and inorganic phosphate close enough to one another to form ATP. So in linear electron flow, electrons move from water to NADPH. Electron transfer is used to create a proton gradient, which then drives ATP synthesis by photophosphorylation. This is one way that the system operates, but there's actually another way that's equally important, what's referred to as cyclic electron flow. In cyclic electron flow, the electrons lost by photosystem 1 reaction center chlorophyll are passed to the primary electron acceptor as before and to ferredoxin. But instead of passing them off to NADP, the electrons are shuttled to the electron transport chain and back to photosystem 1's reaction center chlorophyll, hence the name cyclic electron flow. The electrons cycle repeatedly through photosystem 1 and the electron transport chain. Now what does this accomplish? What's produced? Well, if electrons are not being passed off to NADP, obviously no NADPH is going to be produced. But electrons are still being moved down the electron transport chain. A proton gradient is still going to be created, and ATP is still going to be synthesized by ATP synthase. So cyclic electron flow produces ATP, but no NADPH. Now why is this important? Well, remember that the energy in these molecules of ATP and NADPH are ultimately going to feed into the reactions of the Calvin cycle and be used to make sugar from CO2. As we'll see next, the Calvin cycle requires more ATP than NADPH. To build a 3-carbon sugar, 9 ATP molecules are required, but only 6 NADPH are needed. So cyclic electron flow is a mechanism to bump up the concentrations of ATP relative to NADPH to meet that increased demand by the Calvin cycle. Okay, so now that we've seen how the light reactions capture sunlight energy and convert it to chemical energy in the form of the energetic compounds ATP and NADPH, let's see how that energy gets used to produce sugar. The Calvin cycle enzymatically captures or fixes carbon dioxide to make a 3-carbon sugar, but it's important to understand that a 3-carbon sugar is not produced by taking 3 carbon dioxide molecules and somehow sticking them together to make a 3-carbon compound. Instead, what happens is that single carbon dioxide molecules are captured and covalently linked to an existing organic compound in the chloroplast, a 5-carbon phosphorylated compound called ribulose 1,5-bisphosphate, or RuBP. This compound will then be converted to the 3-carbon sugar glyceraldehyde 3-phosphate through a series of enzyme-catalyzed steps. But if what's being produced is a 3-carbon sugar, then that requires the chloroplast to acquire 3 carbon dioxide molecules to meet the stoichiometry. We're going to go through the cycle, keeping track of the intermediates produced and carbon equivalents at each step. To begin, the enzyme ribulose bisphosphate carboxylase oxygenase, or RuBisco for short, catalyzes the covalent attachment of a carbon dioxide molecule to that 5-carbon ribulose 1,5-bisphosphate molecule, producing an unstable 6-carbon molecule which immediately splits apart into 2 3-carbon compounds of 3-phosphoglycerate. To make a 3-carbon sugar, 3 carbon dioxides would have to be acquired in this way. So we'll note that this is happening 3 times to produce that 3-carbon equivalent. This step is referred to as carbon fixation. Carbon has been captured from carbon dioxide gas and fixed onto an organic compound in the cell as a carboxyl group. This rubisco is arguably the most important enzyme on the planet. Without it, organisms may still be able to capture sunlight energy and move electrons around, but we have no way to convert that to stable organic compounds. So if there's one enzyme that everyone should be familiar with and appreciate, it's rubisco. Next, energy is added using the ATP and NADPH produced by the light reactions. First, phosphate is transferred from ATP to the 3-phosphoglycerate, producing 1,3-bisphosphoglycerate, or 1,3-BPG. That name should sound familiar. It's the same 1,3-BPG we talked about in our glycolysis discussion. In the next step, 1,3-BPG is dephosphorylated and then reduced to form glyceraldehyde 3-phosphate using the reducing power of NADPH. This is called the reduction phase of the cycle. And here's our product, our 3-carbon sugar, glyceraldehyde 3-phosphate, or GAP. These names should all sound familiar. Essentially, what we're seeing here is the reverse of some of the enzymatic steps of glycolysis. There, sugar was being oxidized to extract energy and make ATP. Here, energy is being added back in to form a more reduced sugar. Okay, so let's keep track of our carbons here and see where we are. To produce a 3-carbon sugar, we have to feed in 3 carbon equivalents, or 3 carbon dioxide molecules, each of which gets attached individually, one at a time, to a RuBP molecule. So 3 RuBPs each have 1 carbon dioxide added, producing 3 unstable 6-carbon intermediates that split apart to form 6 3-carbon molecules of 3-phosphoglycerate. Those get phosphorylated to form 6 molecules of 1,3-BPG, which gets reduced to form 6 molecules of glyceraldehyde 3-phosphate, or GAP. For every 3 carbon dioxide molecules we feed into the cycle, the cell can siphon off 1 GAP sugar to use for energy and for building other organic compounds like amino acids, fatty acids, etc. The remaining 5 GAP molecules are used to regenerate the starting substrate RuBP to keep the cycle going. This is referred to as the regeneration stage. Now, this is a complicated set of reactions catalyzed by 7 different enzymes, but we can summarize what happens by just following the carbons. So again, we have 6 GAP molecules. For every 3 carbon dioxides that feed in, 1 GAP gets siphoned off by the cell to use for energy. And we're left with 5 3-carbon sugars to regenerate 3 5-carbon RuBPs. So how is this accomplished? First, 2 sugars are combined to produce a 6-carbon sugar, a C6 compound. So C3 plus C3 gives you C6. That C6 is then enzymatically combined with another C3 to yield 1 C5 and 1 C4 compound. This C4 gets combined with another of the C3s to yield a C7. That C7 is then combined with the last C3 to produce not a C10, but 2 C5s. So did we use all 5 of our C3 gaps? 1, 2, 3, 4, 5. And how many C5 compounds were produced? 1, 2, 3. Okay, so the C5 compound we initially get from these reactions is ribulose 5-phosphate, which is very close to what we're trying to regenerate. We just need to add phosphate. Here's where our additional 3 ATP are needed. They're used to phosphorylate the 3 ribulose 5-phosphate molecules to ribulose 1,5-bisphosphate. And we've regenerated our 3 RuBP. And they're ready to go to be acted on by rubisco to fix more carbon. In case you're interested, this is what that set of enzymatic reactions actually looks like. Here's our pool of 5 C3 gap sugars used in that regeneration phase, which you'll remember can be isomerized to dihydroxyacetone phosphate, or DHAP, another C3 compound. Combine 2 C3s, we get our C6, which is fructose. Combine that C6 and another C3, we get a C5 and a C4. Combine that C4 with another C3, we get our C7. Add a C3 onto that C7, and we get 2 C5s. Those reactions convert our 5 C3s to 3 C5s. Phosphorylation using ATP then regenerates RuBP. Here's another look at the Calvin cycle, adding in a bit more detail so that you can see the structures of the intermediates and how the carbon dioxide gets added in, etc. So what does the cell do with this 3-carbon sugar, GAP? Well, as we just said, it can take GAP and DHAP and covalently link them to form fructose. From there, fructose can be isomerized to glucose. The cell can take a fructose and a glucose and link them together to make sucrose to store for the short term, or it could take glucose and link glucose monomers together to form starch. It's going to vary depending on the type of organism and its metabolic needs at the time. These sugars will then be subjected to the same oxidation reactions we already discussed as needed to produce the ATP to power cellular work. The ATP produced in the chloroplast is used for the Calvin cycle to produce sugar. The cell will then use cellular respiration to oxidize those sugars as needed. It's important to realize that these energy transformations in the chloroplast don't replace glycolysis and cellular respiration. They are the source of the sugar to fuel cellular respiration. And in fact, we can see that not only are the products of photosynthesis the substrates for cellular respiration, but likewise the products of cellular respiration are the substrates for photosynthesis. In the mitochondrion, sugar is oxidized with oxygen acting as the final electron acceptor, and ATP is produced to fuel cellular work, with some energy lost to the universe's heat. As you've seen in this video, the source of that sugar and oxygen is the chloroplast, or photosynthetic microbes. The waste products of cellular respiration, carbon dioxide and water, are combined to form sugar using the energy of sunlight and oxygen as the waste product. In this video, we focused on photosynthesis as it occurs in the chloroplast of land plants and algae. One thing we should definitely recognize, though, is that in terms of global carbon capture, marine phytoplankton, including cyanobacteria and protists, are just as important on a global scale. Fully half of all carbon capture and oxygen production is carried out by these marine organisms, as we can see here in this heat map that illustrates global regions most associated with carbon capture from the atmosphere. The bright red terrestrial regions we see here are where we see the highest levels of carbon dioxide fixation corresponding to terrestrial forests. But all of this blue and green shading over the oceans is illustrating carbon fixation carried out by marine organisms at a lower level, certainly, but on a massive scale in Earth's oceans. And most of this involves an incredibly diverse array of single-celled phytoplankton. So we really have to appreciate that much of Earth's oxygen is being generated by these marine microbes. And this is particularly important to consider in light of the threat of ocean acidification we can expect as the planet continues to experience global warming, and the potentially devastating consequences that's going to have for these organisms and indeed for all life on Earth.